very good afternoon to all. I am Dr. Pooja Malik, Assistant Professor, Department of Agriculture, MMDU Malana, Ambala. Today we will discuss about the seed production of rice. Yes, the rice its scientific name is Oryza sativa, belongs to the family Poaceae and its origin Indo-Burma, having the chromosome number 24. Regarding the seed production of rice, we have some guidelines. The first is land requirement. For the seed production of rice and the land requirement, the same crop should not be grown on the same piece of land for the last one season, unless it is the same variety. If the variety is same, then we can use that land again for the second time and certified by the seed certification agency for its purity. It should be followed for both nursery as well as the main field. Second is the isolation distance. Isolation distance is the minimum prescribed distance between the two crops or between the two different varieties of the same crop to avoid the mechanical mixing or pollen contamination. So, it is specific for specific crops, and isolation distance is more in case of cross pollinated crops and less in self pollinated crops. So, paddy is a highly self pollinated crop, however, some cross pollination does occur. The extent of natural cross pollination varies from 0 to 6.8%. For pure seed production, the seed fields must be isolated by at least 3 meter for both foundation and certified seed production from other varieties and same varieties. As I told that in case of self pollinated crop, the isolation distance will be less as compared to cross pollinated crops, because in cross pollinated crops the contamination chances will be more. So, we have to maintain the more distance to avoid the contamination and mechanical mixing. Secondly, the source of seed. We have to obtain the appropriate class of the seed, either the foundation seed or certified seed, from the source approved by the seed certification agencies. Then regarding the cultural practices, paddy can be cultivated as a direct sown crop, as we can direct DSR or puddle sowing or by transplanting. For seed production, it is desirable to grow paddy under transplanting system so as to avoid the weed problem. Yes, if we go for DSR, direct seeded rice, in direct seeded rice there is a lot of weeds in the field. So, for the production of, if we go for seed production, then go for the transplanting. The seed rate required is 30 to 40 kg per hectare. The spacing adopted 10 to 15 centimeter for early duration varieties and 15 into 15 centimeter for medium sown varieties and 20 into 15 centimeter for late duration varieties. This 10 into 15, 10 means the row to row spacing and 15 is plant to plant spacing. Then transplanting should be done when the seedlings are 3 to 4 weeks old. After 3 to 4 weeks, we can do the transplanting from the nursery to the main field. Then roguing. Roguing of off-type plants should be done once prior flowering, then at prior flowering, second then at flowering time, and third at post flowering, or we can say maturity. Firstly, roguing is the removal of off-type plants from the field, and in off-type plants there is a category of 4 type of plants. Either it may be a different crop plant, secondly it may be a different variety of the same crop plant, it can be a disease plant, and the fourth, it can be a weed seed plant. So, any off-type plant that comes in these 4 categories, that should be removed from the field to avoid the contamination, that is the roguing. So, roguing is done once before flowering, second at the time of flowering, and third at the time of maturity, or you can say at the time of post flowering. The off type should be identified based on the morphological characters such as plant type, plant height, days to flowering, leaf color, flag leaf shape, flag leaf angle, shape of the panicle and color of the glumes. On the basis of morphological characters, you can identify this is the off type or this is the same crop. Rogue out the wild rice plants, plants infested by the stem borer and disease plants such as false mud, paddy bunt. These are the common diseases that comes in the rice crop. Then number of field inspections. The numbers of field inspection required are 2 and they should be done between flowering and harvesting. Why between flowering and harvesting? Because there is a maximum chances of contamination is during flowering and harvesting. During field inspection, the verification should be done for isolation requirement, volunteer plants, off types and disease plants. Then there is some field standards required are as follows: off types, objectionable weed plants, disease plants. Means if we talk about the off type plants, then there is a limit of plants 0.2% in case of certified class seed and 0.01% in case of foundation class. What does this mean? 0.2% means 0.2% means 2 by 1000. And there is a limit of only 2 plants in 1000 plants. If the plants greater than 2 out of 1000 plants, then we will discard that. We will discard that discard that field, because there is a maximum contamination of off types. In the same way, there is a limit of objectionable weed plants 0.02% in certified class and 0.01% in foundation class. Similarly, disease plants also limit is there 0.5% and 0.1%. 0.5% means there is a limit of 5 plants out of 1000 plants, means 5 plants can be diseased in 1000 plants, not more than 5. And for the foundation seed, there should be a limit of 1 plant out of 1000 plant. So there will be more stringency, more strict in foundation seed as well as certified seed. After that, harvesting. The crop should be harvested when the grains become hard and yellow with a moisture percentage of 23 to 24%. For combined harvesting, the moisture percentage should be in the range of 16 to 18%. The crop is cut at the base with a sickle and the plants are left in the field for 2 to 3 days. Then they are threshed on clean threshing floor or tarpaulin. After winnowing and cleaning, the seed should be dried to a safe moisture content of 13% before storage. Then seed yield, the yield in the range of 5 to 6 ton per hectare depending on the variety and the management practices adopted. Then hybrid seed production of rice, how we can produce the hybrid rice. Okay. So, Professor Yuan Longping have the maximum contribution regarding this hybrid rice, and that is why he is the father of hybrid rice in China. The successful development and use of hybrid rice technology in China during 1970s leads the way for development and release of rice hybrids in India. In general, the hybrid rice gives 1 ton more yield than the best variety available. Yes, this is the thumb rule that always the hybrid varieties gives you the maximum yield or 2 to 3 times more yield as compared to the varieties. So, at present more than 10 rice hybrids have been developed in the country from different states, and the first rice hybrid has been developed in the country by Angru University. Then methods of hybrid rice production. Hybrid rice can be produced by 3 different methods. The first method is 3-line system, or we called it ABR line system. In this method, there are 3 different lines, that is A line or male sterile line, B line or maintainer line, and R line or restorer line. Yes, A line is the male sterile line. Here the male is sterile, means non-functional. So, we called A line as a female line because here male is absent or non-functional. So, it becomes female line. Second, B line is the male fertile line because here male is functional. So, we called it male fertile line. B line is male fertile line, A line is your female line. Then R line is the restorer line, that is having the fertility genes to restores the fertility. So, for maintaining A line, it has to be crossed with the B line, as the name pronounced B line is the maintainer line. So, B line, the function of B line is to maintain the A line. Both A and B lines are the isogenic lines. Similar lines, only the difference of a single gene, that is of fertility or sterility, otherwise both are identical lines. So, for maintaining A line, it has to be crossed with the B line, and for producing the hybrid seed, A line has to be crossed with the R line. Yes. So, how we produce the hybrid seed? Using 3-line system. First, A line is crossed with the B line. So, B line, the function of B line is to maintain the A line. Then when we cross A with the B, we will get the A line. Here the A line get maintained. Then after that, A line is crossed with the R line. A line is formed, but it is again sterile. Then to restore the fertility, we have to cross A line with the R line, that is male fertile, and then the hybrid seed is produced. So, in this way we can produce the hybrid seed of rice using 3-line system. Second is 2-line system. This method of hybrid rice seed production involves the use of photoperiod sensitive genetic male sterility system or temperature sensitive genetic male sterile system. In 2-line system, we have two things: photoperiod sensitive genetic male sterility means a particular photoperiod is there, beyond that the plant will behave as sterile, or a particular temperature range is there 23 to 25 degrees Celsius, and beyond that temperature the plant will behave as sterile, and then we can use this 2-line system for the production of hybrid seed. Third is by using chemical emasculates. The chemicals which kill or sterilize the male gamete with little or no effect on the normal functioning of the female gamete can be used to emasculate the female parental line in hybrid seed production. Yes, chemical emasculants, or we can say these are the male sterilants which kills the male plant and creates sterility. And the example of chemical emasculants, malic hydrogides and ethyls, we use this for the production of sterility. Then if we talk about the hybrid seed production, again there is a land requirement is the first step. The same crop should not be grown in the same piece of land in the previous season. Then if we talk about the isolation distance, there is 3 meter in case of hybrid seed production of rice varieties. But if we talk about the hybrid seed production, then the hybrid paddy field should be isolated from the other paddy field by at least 200 meter for the seed classes A class, B class and R line production, and by 100 meters for the hybrid seed production where A with the R line. When we cross A with the R, then there will be hybrid produce. And if space isolation is a problem, then we can go for time isolation or barrier isolation. For time isolation, the difference between the flowering of seed plot and the contaminating plot should be at least 4 weeks. In case of time isolation, we can go for different sowing dates. So, if you go for different sowing dates, then there will be no synchronization of male and female. When both space and time isolation is not possible, then we can go for barrier isolation. What is barrier isolation? In barrier isolation, we use the barrier crops, tall growing crops that are of 6 to 8 feet height size, that should be grown around the seed plot for 10 meters. Barrier crops used like dhancha crop, sun hemp, sugarcane crop and sorghum crop. These crops can be used as the barrier crops in hybrid seed production. Then cultural practices. The success in hybrid seed production depends on the synchronization of the flowering between male and the female parent. Synchronization is a very important term. It means synchronization means the time maturation of both male and female is same. means when the pollen grains are able to dehiscence or they are active to fertilize the female, at the same time the female is ready to receive that pollen grain, only then the fertilization takes place. So, that is the synchronization of flowering between male and female. Generally, the A line is sown once, while the B line or R line is sown 3 times at an interval of 5 days. If A line comes to a flowering in 65 days and R line in 72 days, then the difference is of 7 days. After second sowing of R line, adjust the sowing of A line with a gap of 7 days, so that the synchronization takes place. For example, if first flowering of R line is done on 1st June, second sowing on 5th June and third on 10th June, then sowing of A line should be done on 12th June, so that there will be synchronization of flowering between male and female. Then planting ratio. The commonly adopted planting ratios of male and females are 2:8, 2:6 or 3:8. 2:8, here 2 are the rows of male parent and 8 are the rows of female parent. The maximum the number of female, more will be the production of hybrid seed. So, the maximum number of rows will always be of female, and the role of pollen is only to give the pollen, that is a facilitator, only to give the pollen grains to the female. So, the less number of rows will be of male parent and maximum number of rows will be of female parent, because seed always comes on female parent. That depends on the weather condition and the potentiality of the parental lines. Transplanting should be done when the seedlings are 25 to 28 days old. The spacing adopted for A line is 15 into 15 centimeter. For B line or R line, 20 into 15 centimeter or 30 into 15 centimeter respectively. Number of field inspections. A number of 4 field inspections should be conducted. In case of production of varieties, seed production of varieties, we have 2 field inspection, but if we talk about the hybrid seed production, then the minimum 4 field inspections will be required. First inspection should be conducted before the flowering stage, second and third during the flowering stage, and the fourth before harvesting or at the maturation of the crop. During the first inspection, verification should be done for volunteer plants. Volunteer plants are those plants that grow their own. Isolation requirement, errors in planting and the actual acreage sown. During the second and third field inspection, verification should be done for the isolation requirement, isolation distance, of types, diseased plants, pollen shedders and objectionable weed plants. Pollen shedders are those plants which shed the pollen in different lines, like if we grow the A line in a field, then B line in A line will be the pollen shedder line that spoil the that creates the contamination in A line. So, A line, B line, in case of A line field is the pollen shedders, and the objectionable weed plants are the obnoxious or poisonous weeds in case of main field. Fourth or final field inspection should be done to verify for all the above factors, and the off types can be identified based on the panicles or seed characters. So, we have to identify the off types on the basis of the morphology or phenotypic structures. So, if we talk about the off types, there will be a limit of 0.05% in case of foundation seed and 0.2% in certified seed. Pollen shedder limit will be 0.05% and 0.1% of certified seed, foundation seed and certified seed respectively. Objectionable weed plants will be 0.01% for foundation seed and 0.02% for certified seed. Disease plants will be 0.1% for foundation class and 0.5% for certified seed class. Roguing. During flowering period, roguing should be done daily to remove the pollen shedders from the female parental line. The off-type plant should be identified based on the morphological characters like plant height, plant type, flag leaf shape, flag leaf angle, and disease plants like paddy bunt. Yes, roguing, you have to done the roguing on the daily basis. But if you seen the plant looks different from the main crop field, and instantly at that time rogue out that plant from the field and discard it, so that it can't create the contamination further. The male sterile plants have shriveled anthers and they do not shed pollen, while the pollen shedders have yellow colored plumpy anthers which shed the large amount of pollens. Yes, we can also identify male fertile or male sterile plants in the field. The male sterile plants have shriveled anthers and do not shed pollen, while the male fertile plants have plumpy yellow bright anthers which shed large amount of pollens. Then harvesting and threshing. Harvest the male rows first and remove them from the field so as to avoid the mechanical mixture. Then harvest the female rows. Precaution should be taken while harvesting, not mix male and female plants. Yes, harvest the male and female plants separately. Threshing should be done on a clean threshing floors and the seed should be winnowed and dried to a safe moisture limit before storage. Yes, before storage you keep the moisture content, safe moisture content, so that there will be no contamination after storage. Then regarding yield, depending on the management practices adopted and the potentiality of your parental lines, the seed yield may be 0.5 to 1.5 ton per hectare. Then here we are talking about the hybrid seed production of rice, and we know the rice is a self-pollinated crop. So how we can increase the outcrossing rate, how we can get maximum hybrid production in rice? For that we have some methods that how we increase the outcrossing rate. So paddy is a highly self-pollinated crop and the extent of natural cross pollination is very less. So we have to adopt some natural methods, some methods by the farmer or the breeder to increase the outcrossing rate in rice. So the first method is flag leaf clipping. Flag leaves, here you can see in this picture, this leaf that is shown in the picture, this is the flag leaf. So flag leaves are taller than the panicles, and this is the panicle where the grains will come on the panicle. So the flag leaves are taller than the panicles and are the main obstacles for the pollen dispersal and cross pollination. The flag leaves should be removed or clipped 1 or 2 days before handing, so that it can enhance the uniform pollen movement and wide dispersal of pollen grains to give higher seed set, because it creates an obstacle for the pollen dispersal. So if we clip this flag leaf or we remove this flag leaf, then there will be more seed setting. Second is GA3 application. By applying GA3, we can increase the outcrossing rate. Application of GA3 increases the duration of floret opening. More time the floret will remains open, and stigma receptivity. The stigma will become receptive for more time. So helps in adjusting the plant height of both the parents. Spraying of GA3 should be done twice, first when 15 to 20% of the plants starting heading with 40% of the chemical, and second at 50% flowering with 60% of the chemical. The dose required is 50 grams with knapsack sprayer and 25 grams with ultra low volume sprayer. For first spray use 20 gram GA3 in 50 liters of water, and for second spray use 30 gram in 500 liters of water for GA3 application. And the third and the last method is rope pulling. Rope pulling should be done during the peak flowering time, which helps in shaking the male plants and the dispersal of pollen grains to the female plants. Row pulling should be done daily during peak flowering stage at 8:30 am, and it should be repeated 3 to 4 times a day at an interval of half an hour. So, in this way we can increase the outcrossing rate in case of rice, and we can go for more production of hybrid seed. Thank you so much.